Welcome back to the online class, students. In the previous class, we were discussing about the classification of carbohydrates. Okay, so we know that carbohydrates are classified based on number of sugar units into three types. They were monosaccharides, oligosaccharides, and polysaccharides. And we discussed in detail about the monosaccharides and its classification, and we were discussing about oligosaccharides. In today's class, we are going to discuss about oligosaccharides and polysaccharides. Okay. So oligosaccharides also we have started with the introduction where the oligosaccharides are made up of 2 to 10 sugar units. Okay. So among that, the most common type of oligosaccharide is disaccharides. Okay. We have completed with disaccharides, and so we are supposed to discuss with trisaccharides. Okay. So the next one what we are going to discuss is trisaccharides. Okay. So trisaccharides. So as it is, the trisaccharide is a type of oligosaccharide. Okay. Trisaccharide is a type of oligosaccharide made up of, tri represents 3. So it is made up of 3 sugar units. Okay. So it is a type of oligosaccharide made up of how many sugar units? 3 sugar units. Okay. So the best example for trisaccharide is raffinose. Okay. So example for trisaccharide is raffinose. Okay. So where we can find this raffinose? Raffinose is preferably found in most of the vegetables and fruits. Okay. So as it is a trisaccharide, so which are the 3 monosaccharides present in it? So raffinose is made up of a molecule of glucose, a molecule of fructose, and a molecule of galactose. So all these 3 monosaccharides connected by glycosidic bond forms into what? Raffinose. Okay. The next one is tetrasaccharide. Okay. The next oligosaccharide is tetrasaccharide. So meaning of tetrasaccharide, tetra represents 4 sugar units. So it is the oligosaccharide made up of how many sugar units? It is made up of 4 sugar units. Okay. So example for tetrasaccharide is stachose. Example is stachose. So this stachose is also found in most of the fibrous vegetables. Okay. So preferably it is found in case of beans. Okay. So as it is a tetrasaccharide, it is made up of 4 sugar units. So they are 1 molecule of glucose, 1 molecule of fructose, and 2 molecules of galactose. Okay. So the 2 molecules of galactose connected by glycosidic bond forms into what? Stachose. Okay. So the next is pentasaccharide. Next is pentasaccharide. So penta means 5. So it is oligosaccharide made up of 5 sugar units. Okay. So the example is, it is made up of how many sugar units? 5 sugar units. So example for pentasaccharide is verbascose. Okay. Example for pentasaccharide is verbascose is the example for pentasaccharide. Okay. So it is made up of 5 monomers. They are glucose, fructose, and 3 molecules of galactose. Okay. That is made up of 1 molecule of glucose, 1 molecule of fructose, and 3 molecules of galactose formed by glycosidic bond results in the formation of verbascose. Okay. So all these 3, that is trisaccharide, tetrasaccharide, and pentasaccharides, so they are the part of the human diet, but they are not indigestible. Okay, so they are not digestible. Okay, so it means, so they will not be digested inside the body. But, so what is the function? So it helps in softening of stools. Okay, softening of stools, and not only that, even it helps in the growth of good bacteria in the intestine. Okay, so that is the role of some of the oligosaccharides. Okay, so the next we are going to discuss in detail about polysaccharides. Okay, so the next one what we are going to discuss is the polysaccharides. So as the term suggests, poly means many. Saccharides means sugar. So polysaccharides we can define it as these are the carbohydrates made up of many sugar units which are connected by glycosidic bond. Okay, so we can define the polysaccharide as, so these are the carbohydrates. These are the carbohydrates made up of large number of monomers or sugar units connected or held together by what type of bond? Glycosidic bond. Glycosidic bond results in the formation of polysaccharide. Okay. So the next characteristic of polysaccharide is, so these polysaccharides they are not soluble in water. Okay, and they are tasteless. They are not soluble in water. They are tasteless, and they cannot pass through any membrane due to their large size. Okay, so they are insoluble. They are insoluble in water. They are tasteless. Okay, and they cannot pass through, any membrane. They cannot pass through any membrane due to their large size. The next, so what about on hydrolysis? Okay, hydrolysis, as we know that hydrolysis means breakdown. So these polysaccharides are made up of many sugar units, isn't it? So when they are broken, it forms into many sugar units. That means on hydrolysis, polysaccharides produces many sugar units. Okay. So on hydrolysis, they produce what? Many sugar units. Okay. So this is regarding the introduction of polysaccharides. Okay. Next, we are supposed to know the classification of polysaccharides. So here the classification can be done based on its composition, and other type of classification is based on its function. First, let us see the classification based on its composition. Okay. So classification of polysaccharide. First thing is based on composition. So composition means what they are made up of. Okay. So as we know that polysaccharides are made up of many monomers. Okay. So they are made up of many monomers. Now, so based on composition, we have two types. One is homopolysaccharide, another one is heteropolysaccharide. Okay. So first one is homopolysaccharide. Another one is heteropolysaccharide. Okay, heteropolysaccharide. Okay, first let us discuss about homopolysaccharides. Okay, the term homo represents similar or identical. Okay, so polysaccharide. So it is a type of polysaccharide made up of similar type or only one type of monomer. Okay, so I can define homopolysaccharide as, so these are the polysaccharides made up of one type of monomer. We can call it as homopolysaccharide. So that is the definition of homopolysaccharide. Okay, so let us define it again. So homopolysaccharides are, so polysaccharides made up of, homo means similar, made up of one type of one type of monomer. One type of monomer, we will call it as homopolysaccharides. Okay. Next, let us see the example of homopolysaccharide. So, homopolysaccharides can be, examples are like, so you can take the example of the starch. Okay. The next example is glycogen. Then cellulose, inulin. Okay. So here starch, it is made up of, as I told that homopolysaccharide means it is made up of only one type of monomer. Now starch is a polysaccharide, that is it is a large unit. It is made up of glucose. Okay. So many number of glucose joined with one another results in the formation of starch. Okay. So the starch is made up of glucose. Okay. So can I tell that glucose is the monomers of the starch. Okay. Then next, coming to the glycogen. Glycogen is also made up of, so many number of glucose joined with one another and forms into glycogen. So even in case of glycogen, which is the monomer? It is glucose. Okay. Then next, coming to the cellulose. Cellulose is also made up of glucose. Okay. Many glucose are connected by glycosidic bond and forms into a large molecule called as cellulose. Okay. So next is inulin. Inulin is made up of fructose. So many fructose are connected by glycosidic bond, results in the formation of inulin. So fructose is the monomer in case of where? Inulin. So as we discussed that polysaccharides are tasteless and they are insoluble in water. So but whereas inulin, so it is soluble in water. In some of the cases it has been used in the production of ice creams and all. Okay, and even it is having some sweetness also. Okay, so this is regarding the homopolysaccharide. Okay, next coming to the heteropolysaccharide. So the term hetero represents different. Okay. So different. Poly means many. Saccharide means sugar units. So heteropolysaccharide means what we can call it as? So it is a polysaccharide which is made up of different types of sugar units. Okay. So the definition of heteropolysaccharide is, it is a polysaccharide made up of different types of sugar units. Okay, so that is the definition. It is the polysaccharide made up of different types of sugar unit. Different types of sugar units, we'll call it as what? Heteropolysaccharide. Okay. So let us take some examples of heteropolysaccharide like pectin, lignin, chitin are some of the examples like pectin, lignin, chitin, heparin, hyaluronic acid. So hyaluronic acid, agar. Okay. So peptidoglycan. So these are some of the examples for heteropolysaccharide. Okay. So let us see that. For some of the examples I'll take. For example, like I'll take the example of a chitin. Chitin. So chitin is a heteropolymer. So it is made up of nitrogen derivative of glucose. Okay, that is, we'll call it as N-acetyl glucosamine. Okay, so chitin is made up of what? N-acetyl glucosamine. Okay. The next is, I'll take one more example of peptidoglycan. Peptidoglycan. In this case, peptidoglycan, it is made up of N-acetyl glucosamine plus N-acetyl muramic acid. Okay. So the different units have been combined to form the large molecule like peptidoglycan. Okay. So peptidoglycan is made up of N-acetyl glucosamine. N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl muramic acid. Okay. So then one more example like hyaluronic acid. Hyaluronic acid. So in case of hyaluronic acid, you can find N-acetyl glucosamine will be combined with D-glucuronic acid. Okay. So hyaluronic acid will be combined with N-acetyl glucosamine combined with D-glucuronic acid combined to form what? So hyaluronic acid, which is the one of the example for heteropolysaccharide. So this is regarding classification of polysaccharide based on composition. Let us discuss one more classification of polysaccharides which is based on function. Okay. So another type of classification of polysaccharides is based on function. So based on function, we can categorize them into 3 types. Okay. So based on function, we can categorize polysaccharides into how many types? Into 3 types. Okay. Let us see that. So classification of polysaccharides based on function. Into how many types? Into 3 types. The first one is storage polysaccharide. Storage polysaccharide. Second one is structural polysaccharide. Structural polysaccharide. And third one is mucopolysaccharide. Okay, so based on function, there are 3 types. Which are those? Storage polysaccharide, structural polysaccharide and mucopolysaccharides. So first let us see about storage polysaccharide. So these are polysaccharides which are used as the reserve food material. Okay. So storage polysaccharides are used as what? Reserve food material. They are used as reserve food material. And these reserve food material, when there is a necessity, when there is a need, so these reserve food materials are hydrolyzed. They are hydrolyzed. Hydrolysis means they are broken into simple sugars like glucose. Okay. So these reserve food materials, when there is a need, so they are hydrolyzed. So they are hydrolyzed to produce what? The simple sugars like glucose. So this glucose will be absorbed by the cell. Later in the cell, by the breakdown of glucose, the cell gets the energy. Okay. The cell can get the energy. Okay. So in this way, the storage polysaccharides are used. So which is the example for storage polysaccharide? So the best example for a storage polysaccharide is starch and glycogen. Okay. So one of the example is starch and another one is glycogen. Okay. So first let us discuss with the starch. So starch is the one of the storage polysaccharide found in case of plants. So it is a reserve food material in case of where? Plants. So where we can find? So the whole grains contains the starch, legumes. So like potato, banana, all will contain the starch as the reserve food material. Okay. So this starch is, as we know that the starch is a homopolysaccharide made up of what? Glucose. Isn't it? Actually, the starch is made up of 2 components. One is amylose. Another one is amylopectin. So it is made up of two components. One is amylose, another one is amylopectin. So this amylose is, it is containing around 200 to nearly 1000 or 2000 units of glucose connected by the glycosidic bond. Okay. So then amylopectin, it contains around 2000 to 20,000 units of glucose connected by glycosidic bond. So here amylose, as I told that they are also connected by glycosidic bond. So they will contain 1,4 glycosidic bond. So whereas in case of amylopectin, you can find two types of glycosidic bond, that is 1,4 and 1,6 glycosidic bond. Okay. So what is the 1,4 and 1,6 means? So already we discussed about the glycosidic bond. So where I took the example of, maltose. Maltose is made up of 2 units of glucose, isn't it? So do you remember? So maltose is made up of 2 units of glucose and we wrote like this, isn't it? So glycosidic bond can be defined as, it is a chemical bond formed by the the carbon atoms of the 2 adjacent monomers, isn't it? And we know that the number of carbon atoms 1, 2, 3, 4, 5 and the 6th carbon atom found outside the ring. Okay, so the same follows with another glucose 1, 2, 3, 4, 5 and the 6th carbon atom is on the outside. So now here, preferably for example, I am explaining regarding the amylose. In the amylose, how the glycosidic bond is formed. So it is formed between the first carbon atom with the another glucose of the 4th carbon atom. So I can call it as 1,4 glycosidic bond. So in case of amylose, we'll find only 1,4 glycosidic bond. Okay. So due to the 1,4 glycosidic bond, amylose will have a linear structure. Okay. And due to heavy coiling, so you can find it as a helix. Okay. This linear structure, due to heavy coiling, it forms as a helix in case of amylose. Whereas in case of amylopectin, so it is having 1,4 as well as 1,6 glycosidic bond. Okay. So here what happens in case of amylopectin? So the same thing. So I am writing the 2 sugar units. That is glucose. I am taking it as an example here. So the same follows. The number of carbon atoms are, 5 carbon atoms are found inside the ring. 1, 2, 3, 4, 5, 6th is on the outside. Okay, the same with another 1, 2, 3, 4, 5, 6th will be found on the outside the ring. Okay. Then, in case of amylopectin, what happens? So, one glycosidic bond will be found between first carbon atom of one sugar unit with the 4th carbon atom of the another sugar unit. So, we'll call it as 1,4 glycosidic bond. Okay. The next in case of amylopectin, that is along with 1,4, even we are finding what? 1,6 glycosidic bond. So, here what happens? So, one more glucose molecule will be combining with the 6th carbon atom. Okay. So 1, 2, 3, 4, 5 and 6. Okay. So here the bond is formed between the first and the 6th carbon atom. So glycosidic bond is 1,6 glycosidic bond. Such type of branched structure you can find it in case of amylopectin. So whereas linear structure is found in case of amylose. Okay, is it clear? The next, why I explained you this is because the most common test done for presence of starch is iodine test. When we add the iodine into the starch solution, it produces a deep blue color. So the deep blue color is due to the amylose. So amylose have what? Linear structure, that is, which is called to form helix. When the iodine is added to the starch, what happens here? Iodine combines with the helix structure, or even we can tell that iodine is found inside the helix structure, due to which we get the deep blue color in case of the starch. So this is regarding starch. So the next structural polysaccharide, the storage polysaccharide is glycogen. So glycogen is a reserve food material in case of where? It is found as a reserve food material in case of animals. Okay. Due to that, it is also called as animal starch. Okay. So glycogen. Then next, even the glycogen preferred in case of animals, where you can find the glycogen, they are found in case of liver and as well as in the muscles. When there is a requirement, the glycogen is broken into glucose and it will be used as the energy. So this is regarding the storage polysaccharides. The next one is structural polysaccharide. Okay. The second type is what? Structural polysaccharide. Okay. So structural polysaccharide, as the name suggests. So structural. So it forms into some of the structure, or it forms as a structural framework. So the second polysaccharide based on classification is what? Structural polysaccharide. Okay. So this structural polysaccharide, as I told that, it forms as the structural framework. In case of plant cell, it forms as the cell wall. In case of animal cells, it forms as the skeleton. Okay, skeleton tissue. So in case of plants, it forms as what? Cell wall. In case of animals, it can form as skeleton tissue. Okay. So that is the function of structural polysaccharide. So the most important structural polysaccharide we consider is cellulose and chitin. Okay. So first one I'll take up example for structural polysaccharide is cellulose. The next one is chitin. Okay. So first let us discuss about cellulose. Cellulose, it is made up of what? Glucose. Okay. Cellulose is made up of glucose. Okay. So this cellulose, it is not soluble in water. Okay. Cellulose is not soluble in water, but it can undergo hydrolysis. The hydrolysis of cellulose is done with the help of the strong acid. Okay. The cellulose, it is not soluble in water. Okay. So, but hydrolysis of the cellulose can be done with the help of what? Some of the strong acids. With the help of this, so cellulose can be broken into simple sugars. Okay. And where you can find the cellulose? So, the cellulose can be found in case of cell wall. Okay. Not only in that, so even it is found in the covering of the whole grains. So, you can find the cellulose. So as I told that, it is not soluble in water. It is hydrolyzed with the help of what? Acids. Okay. Next, when you consider not only acid here, even there is one more enzyme called as cellulase. So the enzyme by name cellulase can hydrolyze the cellulose into the simple sugars. Okay. And when you consider the usage of cellulose, the cellulose products are, so it has been useful in producing many different types of products. So preferably, so when you consider cellulose, it is formed as a part of the human diet, as we are consuming some of the plant-based food products. Okay. So even though the cellulose forms the part of the human diet, but it will not be digested in case of humans. What is the reason? Because we are not able to produce the enzyme called as cellulase. Hence, the cellulose will not be hydrolyzed in case of humans. But it is used as a roughage. So, roughage meaning, so it helps in softening the stools and even it helps in the growth of some of the good bacteria. Okay. The next, so even it forms as a diet of some of the other animals like the ruminant cattle. Okay, so it forms as a diet for some of the ruminant cattle which consume the grass and other plant products. Okay, So when they'll consume, in this case, some of the ruminant cattle, the cellulose gets digested. So what is the reason? The reason is there are some of the bacteria, the microbes which are living in the gut of ruminant cattle. Okay, some of the microorganisms living in the gut of the cattle helps in the digestion. Okay, not only cattle, even termites. So you know about termites, right? So termites, they will be attacking the woods, and they'll be feeding on the wood and wood based products and all. The reason is, so in the termite, so the gut of the termite contains some of the microorganisms. Those microorganisms produces cellulose, due to which, so they will feed on the wood or cellulose based products and consume it and digest it. Okay, so this is regarding the cellulose. The next one more structural polysaccharide is the chitin. Okay, so chitin, as we saw, that it is made up of N-acetyl glucosamine. Okay, and this chitin, it is helpful in the formation of exoskeleton of crab, prawn, and even in case of fungi, cell wall of the fungi is also made by the chitin. So chitin is helpful in the formation of exoskeleton. Exoskeleton means it is a protective outer covering. Okay, so skeleton of crab, prawn, and not only in them, even in some insects also they form the exoskeleton. And not only in them, even it forms as a part of cell wall in case of fungi. So it acts as a structural framework in case of fungi and some of the organisms like crab and prawn. So this is regarding structural polysaccharide. The last one is mucopolysaccharide. The third type is mucopolysaccharide. So as the term suggests, mucus. So these are the polysaccharide which form into mucilage-like substance. The polysaccharides which form into mucilage-like substance, will call it as the mucopolysaccharides. These mucopolysaccharides are having various functions. Okay. See, for example, if you consider the one of the mucopolysaccharide called as heparin in case of humans. So it is helpful in inhibiting or avoids the formation of blood clot inside the blood vessel. Okay, heparin avoids the formation of what? Blood clots in case of the blood vessels. Okay, the next is hyaluronic acid. Hyaluronic acid. So this hyaluronic acid is found in case of joints. It helps as a lubricant. Okay, and even some of the examples like agar you can take. Okay, so agar, which is extracted from some of the sea algae. Okay, so it is used as a nutrient medium for growth of microorganisms. Okay, and even it is also used in the industries like in the preparation of ice creams and all. The agar is also used. So this is regarding the mucopolysaccharides. So we are supposed to know the biological importance. So we have studied the carbohydrates in detail, isn't it? So now we are supposed to know the biological importance of carbohydrates. Okay, so how they are useful for the living organisms, or what is their role. Okay, so the next we are going to see what? Biological importance. So biological importance. Okay, so now biological importance. The first one, I'll take the example as, first role is, they are used as a major source of energy. Okay, carbohydrates are used as what? Major source of energy. Major source of energy in case of living system. Major source of energy in case of living system. Hence, even they are called as metabolic fuel. Okay, they are called as what? Metabolic fuel. The best example for that is glucose. Okay, so major source of energy. When we consume the food, it will be simplified into glucose. That glucose will be utilized by your cell. Okay, just like the fuel, so the glucose will be absorbed by the cell, and by using which energy will be produced. That is why we call it as the metabolic fuel. Okay, the next is they are used as the reserve food material. So just now we saw that starch is the reserve food material in case of plant, and glycogen is the reserve food material in case of animal. Okay, so they are used as what? Reserve food material. Are used as reserve food material. In case of plant, it is starch. In case of animals, it is glycogen. Okay. The next is, so even we can find them as structural components also. Okay. Like for example, so you can take the example of cellulose. In case of plant, I am considering here cellulose, pectin, lignin. So these form a part of the cell wall. That means they provide the structure to the cell wall. Okay. So they form the cell wall in case of where? In case of plants. Okay. The next is, so if you consider chitin. So just now we saw that chitin is used in the preparation or in the formation of exoskeleton. Okay. In the formation of what? Exoskeleton. In case of some of the organisms like crab, prawn, and some of the insects use the chitin as the exoskeleton. Okay. So in coming to the animals like, so in case of humans and all, so we can find hyaluronic acid. Okay. So hyaluronic acid and chondroitin sulfate. Hyaluronic acid and chondroitin sulfate. So they form the structural part of the cells. Okay. So that is in case of where? In case of animals. In case of animal, hyaluronic acid and chondroitin sulfate forms the structural components. Then, so we have studied even about the pentoses. In pentose we have ribose and deoxyribose. Okay. So pentoses where we have like ribose, deoxyribose. So we know that ribose is found where? It is found in case of RNA. Deoxyribose is a pentose sugar in case of DNA. That is, so they form as a pentose. Pentose is a type of monosaccharide. They form a part of nucleic acids, which is acting as the genetic material in most of the living organisms. The next is lactose. So lactose, we know that it is a type of disaccharide. Where it is found? It is found in case of milk. Okay. So it is found in case of milk. So this lactose acts as a substrate for, there is a bacteria called as lactobacillus. Okay. So lactose found in the milk act as the substrate for lactobacillus bacteria. So this lactobacillus bacteria feed on lactose present in the milk and convert the milk into curd. Okay, that means, so the lactobacillus bacteria helps in the curdling of milk. So converts the milk into curd. So it helps in curdling of milk, conversion into curd. Okay, not only that, even you can find some more biological importance like agar. So agar is acting as a nutrient medium for growing the microorganisms. Okay, so when we conduct experiment on microorganisms in the laboratory, we require some nutrient medium. So here agar act as the best nutrient medium for growing the microorganisms. Okay, so another example like heparin. Heparin is another polysaccharide, so which inhibits the formation of blood clots. Okay, so it inhibits the formation of blood clots, or it dissolves the blood clots in the blood vessels. Okay, so this is regarding the some of the biological importance of carbohydrates. Okay, so let us see some questions related to this topic. Okay, so here glycogen is stored in, where it is stored? Liver and muscles, only in liver, muscles only, pancreas. Okay, we know that glycogen is stored in case of liver and muscles. Option A is the correct answer. The next, which of the following substance is not carbohydrate? Starch, glycogen, glucose, wax. So, wax is not the carbohydrate. That is the answer. Which of the following sugar is not found in plants? Sucrose, glucose, lactose and fructose. We know that lactose is found in case of milk. Okay, not in case of plants. So, this is regarding today's class. Thank you, students.